A very good morning. I'm P. Sudha Rani, lecturer in microbiology, GDC Man, Srikakulam. Today I'm going to discuss about outlines of oxygenic and anoxygenic photosynthesis. Uh, This oxygenic and anoxygenic photosynthesis, which includes photosynthesis and stages of photosynthesis, the types of photosynthesis, the process of oxygenic photosynthesis, and the process of anoxygenic photosynthesis. The lecture includes all the following things. And as we all know, the photosynthesis is the only way in the universe in which virtually all energy in the biosphere becomes available to living things. And the photosynthesis is the only mechanism which balances carbon dioxide and oxygens on Earth, as well as it is the only mechanism for the existence and sustenance of life on Earth. And this is the photosynthesis mechanism, how it flows. And the photosynthesis is a process by which plants and certain bacteria called phototrophs convert the radiant energy which is derived from the solar energy in the form of light into metabolic energy and reducing power. The photosynthesis includes this is the mechanism of photosynthesis. The photosynthesis includes internal factors and external factors. The chlorophyll, the sunlight is utilizing, as well as carbon dioxide, and the water is utilized in the photosynthesis reaction. And these are the stages of photosynthesis. A photosynthesis mechanism as a whole divided into two phases, the light reaction and dark reaction. The light reaction includes that it traps the light energy from the sunlight and produce ATP and NADPH. Whereas in the dark reaction, the energy which is produced in the light reaction is utilized, and it utilizes the energy from ATP and NADPH, which is from light reaction, and produces glucose. Phototrophs are the organisms which are able to utilize sunlight and produces the carbohydrates. And the phototrophs are divided into two groups based on the evolution of oxygen in the reaction process. They are oxygenic photosynthetic organisms and the anoxygenic photosynthetic organisms. Accordingly, there are two types of photosynthesis processes. They are oxygenic photosynthesis and anoxygenic photosynthesis. Now we'll move into the detail about oxygenic photosynthesis. In the oxygenic photosynthesis, the oxidation of water molecule produces molecular oxygen as a byproduct. Because oxygen is produced, photosynthesis in these organisms is called oxygenic photosynthesis, and the organisms are called oxygenic photosynthetic organisms. Some examples of organisms which undergoes the oxygenic photosynthesis are plants, algae, and some photosynthetic bacteria. And the anoxygenic photosynthesis, which involves a water molecule is not oxidized and oxygen is not produced, and thus the process is called anoxygenic photosynthesis. The organisms which follows this anoxygenic photosynthesis are phototrophic green bacteria, phototrophic purple bacteria, and heliobacteria. This is the reaction of oxygenic photosynthesis and anoxygenic photosynthesis. In the oxygenic photosynthesis, where the carbon dioxide and water, the light energy are utilized, and here we can see the oxygen is liberated. And here in the anoxygenic photosynthesis, where the electron donors are some inorganic or organic molecules, and here we can see that there is no oxygen, and some inorganic or organic molecules are produced in the byproducts. And these are the components of oxygenic photosynthesis. The photosynthesis of oxygenic photosynthesis requires the following machinery for the successful completion and liberation of oxygen, and which includes light harvesting complex, photosystems 1, 2, intermediate carriers, chlorophylls, cytochrome B6F complex, cytochrome complex, quinones, NADP, and FATPase. All this process occurs in the form of jet. Here this is the photosynthesis process, in the oxygenic photosynthesis process, where all the photosystems PS2 and PS1 are arranged, and where the reaction also happens in the form of jet. So it is called as jet scheme. Here we can see the two photosystems work concurrently but in series, such that the flow of electron appears as horizontal jet, hence called the jet scheme. In the presence of light, photosystem 2 feeds electrons to photosystem 1, and the electrons are transferred from photosystem 2 to 1. And the two types of chlorophylls, chlorophyll A and chlorophyll B, participates in the jet scheme, and the jet scheme represents the steps in the light reaction, showing the pathway of electrons transport from water to NADP+. Here we can see the initiating reaction starts from the water, and finally it reaches to the NADP+. This is the overall light reaction where oxygenic photosynthesis, the jet scheme pathway. Here we can see the two photosystems PS1 and PS2. Here the water molecule splits, and it releases the electrons, and the electrons are transferred from water molecule to PS2, and it passes through several intermediates and then finally to PS1, and later it reaches to the NADP, and finally the products are formed, NADPH. And this is the procedure. Now we'll see how the jet scheme is being operated. It requires the electron photon energy, that is from the sunlight. The photosynthesis starts with the simultaneous excitation of pairs of special reaction centers, chlorophyll A molecules named P680 and P700. That is, P680 is the photosystem 2 and P700 is the photosystem 1. The excitation energy comes either from directly absorbed light or by energy transfer from adjacent pigment molecules such as antenna molecules. Like the energy comes either directly from the absorbed light, or the surrounding antenna molecules absorbs the light energy, and through the resonance process the energy transfers from the antenna cells to the pigment molecules, and then the PS2 will excite. The first chemical step happens within only a very few seconds in the reaction of P680 excitation. This is the very fastest reaction in the oxygenic photosynthesis system, where it happens within a very less time, that is picoseconds of time. Here in the photosystem 2, the P680 excites the electron, and it loses the electron and produce the oxidizer compound of P680, and it reduces to pheophytin. The electron from PS2 is reached to the pheophytin, and the pheophytin will be reduced and P680 in the oxidized state. As well as simultaneously the photosystem 1, the P700 also loses the electron, and it will be also in the oxidation state. The two reactions occur in the same simultaneously. The P680 also will be in the oxidized state, and the P700 also in the oxidized state in the simultaneous procedure. The P680, the electron transfer from P680 is reached to pheophytin, and from P700 it reaches to A0. And now the electron transfer steps. The oxidation reduction process drives the activities in the jet scheme in a sequence. The recovery of P680 to P680 oxidized P680 to P680 and P700. The photosystem 1 oxidized state to P700 happens almost simultaneously. The loss of electron also is simultaneous, and the recovery of electrons also occurs in a simultaneous procedure. The oxidized P700 recovers the electron to P700 by receiving an electron that was passed down from the pheophyte into plastoquinone. Here we can see that the electron transfer, the recovery of electron also happens simultaneously from as well as in PS2 and PS1. The loss of electron to PS1 is recovered from plastoquinone. And the quinone B has accepted 2 electrons from quinone A and also takes on 2 photons from the stroma. Then it detaches from its protein binding site and diffuses through the hydrophobic core of thylakoid membrane. Thus the electrons are passed through the intersystem chain of electron carriers. As well as there is a protein complex called cytochrome BF complex, which contains iron sulfur and cytochrome F and cytochrome B6 complex. And here the quinone cycle occurs where the electron transfer happens between the PS2 and PS1, where this is the very smallest reaction in the oxygenic photosynthesis. The electron loss from P680 to the electron loss in P680 is the very fastest reaction, as well as the quinone cycle reaction is the very slowest reaction. It takes several milliseconds. In the PS1, the electron on A0 is passed ultimately to NADP plus via several other intermediates like A1, phylloquinone, and FX, FA, and FB, which are immobile iron sulfur proteins. And the missing electron on P680 is also recovered ultimately from water molecules, and these reactions happens. Here we can see the loss of electron also recovered from the water molecules. The loss of electron, which is by through the photons, where the PS2 is lost this electron, and it is recovered through the split of water molecules. Now we'll see the statistics. What is the loss of electrons and how much quantity of energy is lost and how much quantity of energy is utilized in these reactions. A total of 8 quanta of light, that is 4 in photosystem 2 and 4 in PS1, are required to transfer 4 electrons from 2 molecules of water to 2 molecules of NADP+. This produces 2 molecules of NADPH and 1 molecule of oxygen. This is the oxygen that both the plants and animals need for respiration and life to occur. Now we'll see not all organisms utilizes the same machinery and same kind of photosynthesis. There are some variations in the oxygenic photosynthesis. The evolution of oxygen mechanism is similar in algae, cyanobacteria, and in higher plants, but the presence of photosynthetic pigments, associated proteins, as well as antenna proteins also vary among these 3 groups. In higher plants and in algae, both chlorophyll A, chlorophyll B participates in the jet stream reaction, but in cyanobacteria, chlorophyll A only participates in the reaction. This is the major difference among all these organisms which follow the oxygenic photosynthesis. And the second kind of photosynthesis, anoxygenic photosynthesis. Anoxygenic photosynthesis, which we dealt previously, like where oxygen is not liberated in this photosynthetic reaction. Anoxygenic photosynthesis is the phototropic process where light energy is captured and stored as ATP. The production of oxygen is non-existent, and therefore water is not used as an electron donor. This phototropic process derives its name from the fact that water is not used as an electron source, and therefore oxygen is not produced, and so no oxygen will be liberated in this reaction. Anoxygenic photosynthesis depends on electron donors such as reduced sulfur compounds, molecular hydrogen, or organic compounds. Here we can see the reaction. The organisms that follow this anoxygen photosynthesis are mostly found in freshwater, brackish water, marine and hypersaline water. But here we can see in the reaction like carbon dioxide plus some inorganic or hydrogen sulfide or some inorganic or organic molecules participates in the reaction, as well as light also are useful for this anoxygenic photosynthesis, where the glucose is formed, as well as some inorganic molecules or byproducts are formed in the anoxygenic photosynthesis. Now we'll see the what are the organisms follow this anoxygenic photosynthesis. Mainly three groups of bacteria carry out anoxygenic photosynthesis. They are phototrophic green bacteria, phototrophic purple bacteria, and heliobacteria. There is some difference in the oxygenic and anoxygenic process. Unlike oxygenic photosynthesis, the process involves participation of only one photosystem. There in the oxygenic photosynthesis, where we have seen that two photosynthesis participated in the oxygenic photosynthesis, they are arrayed in the same line, and the electron transfer occurred in the jet scheme. Here we can see that only one photosystem participates in the anoxygenic photosynthesis, either PS1 or PS2 participates in this photosynthesis. In some bacteria, the photosystem is similar to PS1, and in some others, it is similar to PS2. And these are the machinery which are required for the conduct of anoxygenic photosynthesis. They are photosystems, bacteriochlorophylls, inorganic molecules, intermediate electron carriers, quinones, cytochrome B6, BC complex, etc. And this is the flow of electrons. Here we can see, as we have discussed previously, there is only one photosystem participated in this anoxygenic photosynthesis process. Here we can see P870 is the photosystem participated in the anoxygenic photosynthesis. Here in the down we can see that the light energy is when the photon quantum of energy passed through this photosystem, it excited. The excited state of photosystem, the electron from the excited photosystem is transferred to bacterial chlorophyll, bacteriophytin, and to several intermediates, and finally reaches to cytochrome BC1, cytochrome C2, and again through external electron donors, the electron is again the depleted electron is again transferred to the P870. And so there is no loss of electrons here, because the electron is again transferred to P870. Now, after looking this, now we'll see the variations in anoxygenic photosynthetic organisms. Here some variations. The purple and green bacteria utilize bacterial chlorophyll and hydrogen sulfide, S8, S2O3, hydrogen, ferrous ions, and organics as electron donor. In the purple non-sulfur bacteria utilize hydrogen as their reducing agent, and they do not release sulfur. The heliobacteria are the anoxygenic phototrophs that contain bacterial chlorophyll G as their sole chlorophyll pigment, and the nitrogen fixing heterotrophic organisms found in soil and rice paddies. And these are some variations in the anoxygenic photosynthetic organisms. Now, finally, we'll see what are the differences of oxygenic and anoxygenic photosynthesis in a glance. At a glance, we'll see now. As we know, oxygenic photosynthesis is the photosynthesis that occurs in plants, algae, and cyanobacteria, in which the final electron acceptor is water, which we have seen in the reaction. The final electron acceptor is water. But in case of anoxygenic oxidants, the case is different. Photosynthesis is a form of photosynthesis used by certain bacteria in which oxygen is not liberated. And the organisms which utilizes this oxygenic photosynthesis are plants, algae, and cyanobacteria, whereas it is completely different in anoxygenic photosynthesis. Green sulfur bacteria and non-sulfur bacteria, purple bacteria, heliobacteria, and some acidobacteria follows this anoxygenic photosynthesis. Photosystem 1 and 2 are used in this oxygenic photosynthesis, whereas in case of anoxygenic photosynthesis, only one photosystem is used. Water is the sole electron source in the oxygenic photosynthesis, which splits the electrons the water molecule splits and it supplies the electrons to the photosystem 2 and through the PS1, whereas in case of anoxygenic photosynthesis, no water is utilized. Hydrogen, hydrogen sulfide or ferrous ions serves as the electron donors for this anoxygenic system. Oxygen is produced during the light reaction, where in an oxygenic photosynthesis, oxygen is not produced. And the chlorophylls are used, different chlorophylls, chlorophyll A, B, and D are used in this oxygenic photosynthesis. But in case of an oxygenic photosynthesis, only one kind of chlorophyll and sometimes bacterial chlorophyll are used. ADP serves as the terminal electron acceptor, producing NADPH by utilizing the terminal electron acceptor is NADP plus also, and with the energy generated from ATP, and it utilizes the ADP and NADP plus and produces NADPH, whereas NADPH is not produced as the electrons or because the electrons, as we have seen previously, the electrons are cycled from back to the system, so there is no transfer of electrons to other components or electron carriers. And finally, ATP is produced by non-cyclic photophosphorylation and ATP is produced by cyclic photophosphorylation. Thank you.